Started. Thank you for joining everyone. I'm Megan Weber. I'm an aquatic invasive species extension educator here at the University of Minnesota. And I am excited to have Daryl Gohl joining us today as our guest speaker on genomic surveillance of zebra mussel populations using genotyping by sequencing. I have a couple of housekeeping items to get started, and then I'm going to turn it over to Daryl for the presentation. We will be using a tool called Slido for our Q&A today. There, you can go to it at any point by going to slido.com and entering the code mussel-strains. And that will get you into the Q&A. If you have troubles using it, don't worry if something ends up in chat. We'll make sure that it finds its way over there. And we are recording today. So if you have to leave early or miss something and want to reference it, we'll have that up on our YouTube channel a little bit after the webinar closes. It just takes a little time for transcription. Everyone does remain muted for the duration of the webinar. So if you have questions, make sure that they go into that Q&A tool. And if you're having technical issues or anything along the way, feel free to drop those into chat. My colleague Michelle is on behind that AIS Detectors program name, and she will do her best to help with any troubleshooting that she's able to do on her end. That's all that I have for our housekeeping type items. So I'm going to turn the mic over to Daryl, and we can get started on the presentation. Thanks so much for joining us, Daryl.
Thank you, Megan. All right, I'm just going to share my slides here. All right, hopefully people can see that okay. All right, well, so my name is Daryl Gohl. I lead a lab here at University of Minnesota in the Genomics Center. So we work a lot on DNA sequencing technologies. Um, Joining me here is a postdoc in my group, Victor Hernandez, who has contributed to this project, and he's working on some new tools and methods that he's going to also talk about later in the talk today. And then I also want to acknowledge Dr. Mike McCartney, who can't be here today. He's out on vacation out in Yellowstone, but he really spearheaded this project and continues to lead these efforts. And he has a deep background in invasive species population genomics. So um, I'm talking sort of in his stead for a lot of this project, in particular the publication I'm going to be telling you about that we published last December. So what is genotyping? So I'm sure people here have heard of services like 23andMe and Ancestry.com. These are companies where you can send them a sample of your saliva, and they'll characterize a large number of genetic markers throughout your DNA. And then that information can be used for a variety of purposes. So one of which is tracking ancestry or understanding sort of what geographical regions your DNA is most associated with. People also use this information to identify long lost relatives, or it can also be used in forensics. So increasingly, law enforcement is using the information that's in these public databases to, solve cold cases. And so what we're doing in this project with zebra mussels is very conceptually similar to what these services do. And I sometimes jokingly call this 16andMe because zebra mussels have just 16 chromosomes rather than the 23 that humans have. Um, so how does one actually go about genotyping? So the way that one way to do it is to sequence the DNA. And one of the issues with just sequencing the zebra mussel genome for all of these samples is that these genomes are very large. So the zebra mussel genome, the diploid genome, is about 3.6 billion letters long. And so just to give you a sense of how much information that is, if you took the entire Harry Potter series and stacked up 700 copies of the series, that would basically be about 3.6 billion characters. So about the length of the zebra mussel genome, and that would be a stack of books, roughly the height of the IDS tower in downtown Minneapolis. And to actually, in practice, to sequence the genome and to get kind of reliable base calls at each position, you actually need to sequence each position multiple times. And so the gold standard, if you're sequencing a human genome for disease diagnosis, would be to get about a 30x coverage of that genome. And so the amount of information to actually kind of in practice genotype by whole genome sequencing is even larger than that. That it's about the equivalent of 21,000 copies of the Harry Potter series or a stack of books taller than Mount McKinley. So in order to kind of do this on a large number of samples in an economical way, we turned to a technique that's part of a broad family of DNA sequencing approaches called reduced representation sequencing. And we're using the specific approach that we're using is referred to as genotyping by sequencing. Um, this is a way of taking that very, very large 3.6 billion base pair genome and selecting out small fragments of it that we then can reproducibly look at in multiple mussels. And so we're taking this snapshot of just portions of the genome with informative genetic markers as opposed to the whole genome. And so in practice, what you do is you take an enzyme called the restriction enzyme. It cuts at specific positions in the genome that give you these small fragments. You then sequence those fragments and then can look at the alignment of those fragments to the reference genome. And so what I'm showing here are you know, several different samples. Zoom in on some of these fragments aligned to the reference genome. And anything in gray basically matches that reference sequence, whereas the bases that are indicated here, the T's and G's and C's and A's, those would be deviations from the reference sequence. So variants that are unique to to that specific sample, um, or not maybe not unique, but differ from the reference sequence. And so these are referred to variants or sometimes single nucleotide polymorphisms or SNPs. So you might hear me using those terms interchangeably throughout the talk. Um, and what you can see from this, just looking at these two little regions of the genome, sample one looks very similar. It has these three changes that are shared with sample three, whereas samples two and sample N here look different. And so by doing this across many, many regions of the genome, you can build up an idea of how related or different and any two samples are. So to start today, I'm going to be telling you about a study that we just published back in December. And as I mentioned earlier, this was led by Mike McCartney um, with some contributions from Victor and I and some others. And the goal of the study was to really look at the early stages of the zebra mussel invasion here in Minnesota and see if we could use these genetic markers to trace the mussels from specific lakes back to their source populations in a very similar way to what Ancestry.com would do when they tell me that I have ancestry in Europe, in Germany, say. Um, and so the collections for this study focused on uh, basically lakes that were infested prior to 2018. And so this is a plot that Mike made uh, of infestations in various states all throughout the Great Lakes region. And so what you can see is that Minnesota was delayed relative to New York and Ohio and Wisconsin, some of these other states that um, share more shoreline with the Great Lakes. So our infestation kicked off later than some of these other states. What you can see is it's in a period of rapid expansion, and this curve is continuing to sort of rise dramatically. And the period that we're looking at is really from the very, very start of the invasion um, through this period of initial rapid expansion into the state. The samples that were collected for this study come from a variety of lake-rich regions throughout the state, and these are somewhat sort of arbitrarily demarcated here with geographical identifiers that I think most people in Minnesota would at least find fairly intuitive into these um, colored lake-rich regions. And so Mike and his postdoc Sophie Malaz traveled around and collected a large number of samples from throughout the state. These encompassed 82 water bodies, 61 inland lakes, almost 2,000 total samples that were then run through this genotyping by sequencing process and genotyped at high resolution. In addition, Mike and Sophie collected samples from throughout the Great Lakes, and you know this is a very large geographic area. Um, and so it's relatively sparsely sampled, but we do at least have representative collections from several points throughout the Great Lakes, including Lake Superior and the Duluth Superior Harbor, which is the area that shares direct shoreline with Minnesota. And so when we carry out this process of isolating out these small regions of the genome and sequencing them and looking at the genetic markers, this is a map of where we find these variants along the 16 chromosomes of zebra mussels. So these chromosomes are numbered in order of basically the biggest chromosome to the smallest chromosome. You kind of see the outline of those chromosome lengths in this map. And then those genetic markers or single nucleotide polymorphisms are shown as ticks on this plot. So you can see these are spaced widely throughout the genome. They cover all 16 chromosomes, and they give us a kind of a good representation of what the genetic background of a specific animal is. So what this ends up doing then is when we actually go ahead and sequence all of these mussels, we end up with this very rich data set that has thousands and thousands of genetic markers. So each row in this large table is a different genetic marker, and each column represents an individual mussel. And so this is a large multi-dimensional data set. And so it's not easy to sort of compress all this data down and visualize it in a way that humans can make sense of. And so we use a variety of what are called dimensionality reduction techniques, and these are essentially mathematical algorithms that take something in a higher dimensional space, like these colored squares here that are shown in 3 dimensions, and you can take that information and basically compress it down into a lower dimensional space that you can more easily visualize. And so rather than doing this with 3 dimensions here, we're doing using a similar kind of mathematical approach to do this with 5,000 plus dimensions, and then compressing it down to 2 or 3 dimensions where we can more easily see what's going on. And so one such dimensionality reduction technique is this principal component analysis. And so what you're seeing here are basically each data point represents an individual mussel, and the spacing between those points essentially are taking all of those 5,000 data points from each mussel and giving you effectively sort of a distance between the different mussels. And so what immediately jumped out from looking at this plot is that within the Great Lakes and the Mississippi River, there are 3 really distinct groups of mussels. And we interpret that to mean that when mussels were first introduced into the Great Lakes, it's thought that they were brought over from Eurasia in ballast from ships. And this suggests that there were basically likely 3 independent events where mussels were introduced into the Great Lakes. There are 3 very genetically distinct populations. And then that genetic diversity in the Great Lakes has seeded and formed the basis of all of the other diversity that we're seeing throughout the country in inland lakes. So another way that we can take and reduce down those thousands of data points to something more manageable that we can visualize is through this sort of admixture analysis. And so what I'm showing here are ancestry plots or Q plots that essentially define specific genetic clusters that are represented across large numbers of mussels and that kind of co-occur. And so each of these different colored bars in these plots represents one of these nine genetic clusters. And each column in these plots is an individual mussel. And so if you look at, say, like the Pelican Lake samples in the upper left-hand corner, you can see that, basically all the mussels in Pelican Lake have this green uh, cluster two popular or set of SNPs, and then other SNPs from cluster eight. And so there's a real kind of clear signature based on these ancestry clusters, and different lakes and different regions have different combinations of these clusters of ancestry. So we can do this and kind of look at all of the different regions, and these plots give you basically an intuitive idea of which lakes are likely to be related to one another. So you can see in this Brainerd Lakes region, for instance, there's this cluster of mussels from Rice Lake and Pelican, Round, Lower Hay, North Long, and Gilbert that all look pretty similar, whereas the mussels from Gull and Pelican Brook and Ossawinamakee look different. So these plots start to allow us to generate hypotheses about which lakes may have been responsible for infesting which other lakes, or at least which ones are closely related. Um, and then we also have similar data for the Upper Mississippi River and its tributaries and the other Great Lakes. And so once we have these ancestry clusters, what we can then do is start to model and do formal testing of hypotheses that we can generate based on looking at these data. And so I'll take you through how that was done quickly. So the first lake that was infested, the first inland lake infested in Minnesota was Ossawinamakee in 2003. And at that time, there were limited number of potential source populations for infesting Ossawinamakee, right? So there were the Great Lakes, which had been infested throughout the 80s and 19 or early 90s, and then there was the Mississippi River. Um, and so looking at the data for Ossawinamakee, you can see that it's composed of this green cluster and this grayish cluster, which looks a lot like the Mississippi River and also a little bit, also a lot like the mussels from the Duluth Superior Harbor. So those were sort of the two most likely candidates, and you can see they look very different from Ossawinamakee looks very different from Lake Michigan and the other Great Lakes. And so what we then do is we can build these trees that and place Ossawinamakee at different points on this tree, and then essentially statistically test how well these different scenarios are supported by the genetic data. And the winning scenario that we get is this one, which shows that the Mississippi River was infested from mussels out of Duluth Superior Harbor, and then subsequently the infestation in Ossawinamakee came from the Mississippi River. And it's fairly high confidence. So the way that these models work is they actually use a sort of machine learning random forest model that assigns different numbers of votes to these different scenarios, and then can calculate a probability of how likely this scenario is given the data. These scenarios can be more complex. So this is just showing one for Cass Lake. So Cass was actually infested in a what's called an admixture event. So essentially mussels from two different lakes likely infested Cass, and then those populations mated and intermixed. And the two lakes that were identified were um, for Cass were Ossawinamakee and then also Lake Erie. Now, there's a bit of a caveat to this analysis, which is that there are a lot of intermediate lakes throughout Wisconsin or throughout Michigan or Illinois that we haven't yet sampled, and so it could be that a boat came from Ossawinamakee and a separate boat came from Lake Erie, or it could be that there was some intermediate lake between Lake Erie um, and Cass that was part of that chain of infestation. And so that's an important caveat to keep in mind when we talk through these scenarios is that our ability to kind of assign the source populations is only good as our database of those potential source populations. Um, but then we can then also in this particular scenario we're testing where the infestation of Winnebagashis, which then looks like it came out of Cass Lake. So just kind of zooming out to this large data set that we have, um, there were some early infestations in Minnesota that came from out of state. So look like mussels that came from either Lake Erie or Lake Michigan. And again, with the caveat that this could have either been one large long hop or there could be an intermediate hop or multiple hops in between. Um, and then we actually see the majority of the early invasions in Minnesota came out of either Lake Superior or the Mississippi River. And I'll just note that these are all shown as a particular point in the Mississippi River. We've sampled mussels from throughout the Mississippi, and uh, they're not really differentiated from one another. So these mussels didn't necessarily come from southern Minnesota. They could have came from anywhere along the length of the Mississippi. But then after these initial sort of longer range hops, we saw a lot of local spread. So this is just showing a few different lake-rich regions. So within Detroit Lakes, we saw that Lizzie was responsible for the infestation of a number of nearby lakes. Same with Pelican. Alexandria, we saw a complex picture, but again with a lot of kind of long jumps and then local hopping in the immediate vicinity. And similarly in Brainerd, and this was actually a little bit surprising because we saw Rice Lake was the which is contiguous with the Mississippi River, was the first area to be connected, and this actually was responsible for the spread into a number of nearby lakes, which is surprising because this is not a lake that shows up as having particularly high boat traffic or connectivity, but nonetheless, it's seeded a number of infestations in the Brainerd Lakes area. So one of the other things that we did in this paper, or that Mike did really, is compare the information about boat traffic or using either data from the Minnesota Watercraft Inspection Program from the DNR or from an app that tracks connectivity between lakes based on anglers reporting their fishing information. Um, and what we saw is that there was not a very strong correlation between the amount of boat traffic in a lake and the number of infestations that lake was responsible for. So you have some high traffic lakes that led to a number of infestations, but a number of the lakes that were sort of responsible for the most infestations were either moderate or low traffic. So the spread or the traffic between lakes does not necessarily explain the spread or what makes a lake particularly susceptible to spreading lots of mussels. Um, I guess I'll just mention one other interesting story from the paper, which is that within the Alexandria region, we actually saw distinct genotypes between a number of lakes that are actually interconnected like internavigable. For instance, Geneva and Lahamadou looked very different from Lake Carlos, despite the fact that these lakes actually, you can boat between all of them. And so this could be a function of the fact that these lakes got independently infested from different source water bodies. And, at the point at which these samples were connected, those mussels hadn't yet had time to interbreed. Um, but it'll be interesting to follow up with those populations and see if they continue to remain genetically distinct or if they start to homogenize over time. So just to summarize a few of the key insights from the paper, we showed that the North American zebra mussel invasion was likely seeded by three separate events in the Great Lakes. That Within lakes, the populations are actually very genetically homogeneous. So looking, we had 60 some samples from Mille Lacs Lake, and they all look very similar, at least within that lake. The exception to this was a couple of the Great Lakes, which within the Great Lakes, there are a couple of areas where there are genetically distinct populations at different, distant areas in the lakes. Um, and then the patterns of spread are generally characterized by a jump from a distant location, followed by a lot of local dispersion. And finally, there's a mixed degree of agreement between what one might expect from patterns of recreational boater traffic and the genetically defined source populations that we were able to characterize. So that sort of summarizes our initial study. And then we've been now working to extend on that work. And we received a grant about a year and a half ago from the USGS to basically do more of this. So the goals of that grant were first to develop methods for genotyping low biomass samples like zebra mussel veligers. Um, and one of the issues is that early on in infestations, often one doesn't find adult mussels in the lake, and so being able, but people will identify like in plankton toes these small larval stages of zebra mussels called veligers, and so being able to apply this tool to those veligers would let us more quickly identify the source population for a given lake. So part of this grant is focused on methods development. That's what Victor is going to be telling you about. And then we're also working on expanding these efforts. So sampling some of the 100 plus lakes that have been infested in Minnesota since 2018, and also expanding geographically and trying to fill in some of those potential missing links so that we can better more reliably trace the source populations. Um, and then finally, the third goal is to develop a web-based interface for exploring this data so that members of the public or uh, invasive species managers can look at this information and potentially use it to help inform, say, inspection efforts. And so I'm going to turn things over to Victor, who's going to be telling you about his efforts to develop methods for genotyping zebra mussel veligers.
Right. Hi, everyone. My name is Victor Hernandez. I'm working with Daryl and Mike on this beautiful project. As Daryl already mentioned it, we have genotype about 2,000 mussels with GBS. And this technique is highly efficient and tailored to large scale studies. It delivers consistent results across samples and tracks thousands of genetic markers across large populations. However, this technique requires high quantities of genomic material, and it is inefficient with low input DNA, which leads to poor library preparation for sequencing. And this precludes its direct use on low biomass samples, such as zebra mussel beligars. So we have proposed multi-displacement amplification, MDA, for increasing the zebra mussel DNA concentration in low biomass samples to be able to genotype genetic markers with GBS. So in short, MDA uses a 529 polymerase and random hexamers to generate multiple copies of the genome from very few nanograms of DNA. Now we have refined an MDA protocol to obtain functional and high quality DNA from very few nanograms of DNA. We have found that the total amount of DNA that can be isolated from a single veliger ranges around 1 nanogram. With this amount of DNA, our protocol yields more than 2,500 nanograms of DNA with a minimum reaction time of 10 hours, thus overcoming the input mass limitation. In the following slides, I'm going to show how we evaluate the quality of the amplified DNA. First, we determined the effect of MDA on whole genome sequencing of zebra mussel DNA. For this particular experiment, two samples from mussels collected in Minnetonka and Turtle Lake, where the DNA were diluted to 1 nanogram, treated with MDA and sequence, and analyzed along with the corresponding controls. And the samples that we treated with MDA, shown here in blue, displays good sequencing quality scores. And also the percent of map reads for these samples was bigger than 90%, which means that most of the reads or most of the data generated in this sequencing experiment aligned with the reference genome. And lastly, for the genotypic data, we identified no big differences in the variant count numbers of the samples that we treated with MDA. So then we used genotyping by sequencing to generate data for our MDA samples, and we used 3 different zebra mussels populations. We analyzed the DNA from mussels from Duluth, the Illinois River, and Lake Erie, and treated with MDA and GBS, as I mentioned. And here, for the genotyping data that we generate in this experiment, we calculate the genomic distances, where the numbers close to 0 in this image represent a bigger chance of genetic similarity. And our samples, or MDA samples marked in blue here, form compact clusters with their corresponding controls, and were assigned to the right source population. So taking into account our GBA or whole genome sequencing and this data from diluted DNA, MDA seems to be a promising tool for low biomass samples. And so we assessed this with actual veligers from Minnetonka, and we used adults from the same location as a reference population. And as expected, our MDA samples or the MDA treatment didn't affect the sequencing quality. We were able to estimate the genomic distance between the control groups and the treated populations. And we observed clustering between the Minnetonka and the veligers from the same location. And these clusters had lower internal genetic distance. And also these samples were separated from Duluth, Illinois River, and Lake Erie. So far, we were able to generate genotyping data comparable to the adult reference population, and this is quite good. And finally, we will continue to work on this project to establish a veliger collection from diverse geographical locations. We will continue with the validation of our protocol, and our goal is to make this accessible for environmental managers. So
All right, great. Thank you, Victor. Yeah, I think it's very exciting that we're very close to, I think, being able to apply this technique to start genotyping veligers in addition to adult mussels. So we're also working, as I mentioned, to expand this work geographically and temporally. Over the last year or so, we've gotten mussels from over 75 additional lakes, mostly here in Minnesota, but also starting to work with external collaborators to get mussels from other states as well. Um, Here, I want to especially thank our partners who've assisted with the collection, which has included both or citizens as well as local, state, and federal agencies. And we've gotten special help from the Minnesota DNR. So I really want to thank them. We're working with the watercraft inspection program this summer to collect more samples. We've had a number of invasive species specialists throughout the state who have really gotten us really great collections from specific regions. I have a QR code here for a wish list and sample form. So if you can go there and actually browse which lakes in the state we still need to collect from. And if you are able or you have access to mussels from that lake, you can fill out that form and sign up, and we'll get in touch with you about getting mussels from your lake of interest. Okay, so the final thing that we're going to talk about is this web-based interface for exploring the zebra mussel genotyping and source population data. And so we've been working together with Alex Badges, who's a quantitative ecologist with MASERC, and Alex has developed a number of web-based tools for exploring AIS data that many of you may have used for other content in other contexts. So we've taken the data that we published in this recent paper, and we've developed a website which we're calling Mussel Strains. That pun is all Alex's, but I think it's a fun little name for this tool. Um, this is what the website looks like, and there again, there's a QR code and link here if you want to look at that website and explore the data. I think we'll also be putting the links to the paper and the collection form and this Mussel Strains app in the chat as well. So I'm just going to give a brief little walkthrough of how we're designing this tool and what you can do with it. Um, so when you go to the Mussel Strains kind of homepage, you'll get a visualization of basically all of the lakes, all of that source data, which you can see is a very busy mess of arrows for all these different movements. You can also see the ancestry plots, and you can scroll down and sort of see the ancestry for all of the different lakes that are being shown here. And then we also have a variety of filters that you can apply to the data. So, for instance, you can click on filter by invasion year, and there's a little slider where you can look within a specific time frame. And if you click this regenerate outputs button, it will apply that filter. And so this is now showing the data just for the final 3 years of that initial data set this would be lakes that were infested between 2015 and 2018. And so you can see the arrows become much more manageable. You can kind of see what's going on a little bit better. And then these maps are explorable. And so you can then sort of click on these plus minus arrows and drag the map around and zoom in on the specific regions of interest. And so this is sort of an example here showing a lake that was responsible for 4 different infestations in this time frame, right? And so what we're envisioning is that as we continue to add data and update this, this could be a very nice tool for identifying lakes that are sort of causing a disproportionate amount of the spread, and potentially a tool for managers to, for instance, if you identify such a lake, to apply more inspection resources there, or take other preventative measures. Um, so with that, I'd just like to thank all of our collaborators. I'd especially like to thank Mike McCartney, who really pioneered and continues to spearhead this work. I'd like to thank Alex Badges, who has done some really great work putting together that Muscle Strains app. And then all of our collaborators here, both in my lab and the Genomics Center at the University of Minnesota. I'd like to thank our funders as well. So that this project was initially funded by state funding from a grant from MASERC and the Environmental and Natural Resources Trust Fund. And then, as I mentioned, we've subsequently gotten funding from USGS to continue and expand this work. So with that, I'm happy to take any questions, and I'll just put up for the moment this slide that has both contact info for me and Mike, and also those QR codes that will take you to these different resources.
Great. Thanks so much, Daryl and Victor, for sharing all that you had. Um, I just got a note from Alex for those of you who are having troubles accessing the Muscle Strains web app, I think the host service was not ready for the number of clicks that it got from that link. So it may have overloaded. Alex just did some work in the background. And if you refresh, you may be able to get in now. I think it has some added capacity to handle the webinar now.
Okay. Otherwise, yeah, check back later when traffic will be lower, I guess.
Great. All right, I'm going to take over the screen share again real quick. So as I mentioned earlier on, we're using a tool called Slido for our Q&A. The great thing about Slido is you can upvote questions. So if you see questions in there that you really want to be answered, you can give them a thumbs up and it moves them up towards the top of the list, so they're more likely to get answered in the time that we have. You can use the QR code showing on the screen now to scan and get directly in there. Otherwise, you can go into a web browser and go to slido.com and enter the code mussel-strains, and it'll get you into that Q&A. If for some reason Slido isn't working well for you, if you do put your question into chat, Michelle is there to move it over and make sure that it ends up in Slido so that I see it as I am working to moderate the questions coming in. So with that, I'm going to go ahead and get started with the first question, Daryl, which is wondering if you've done any genotyping yet on sources from Europe or Asia.
No, we haven't yet, and we would like to. There was a nice paper published several years back by a group of scientists in Russia that uh, collected samples all throughout Eurasia, both the native range of mussels, or of zebra mussels, and also their invaded ranges throughout Europe and Asia as well. And so we know of at least this very, very comprehensive bank of samples that exists in the Russian Academy of Sciences. Um, we're not sure if we'll be able, there are obviously geopolitical reasons that it might be difficult to get our hands on those samples. And then, of course, the native range of zebra mussels is also, around Ukraine. And so there are obviously issues with collecting there at the moment. So the short answer is we have not yet, but we would really like to, and we're hoping that we can maybe find a way um, to start a collaboration with some of these scientists that have already gotten nice sample sets. And I'll just say the work that they did on the samples that were previously collected was an approach called microsatellite marking, or sorry, microsatellite. They use microsatellite markers for genotyping, which is a lower resolution approach. And that's actually what Mike McCartney had started doing initially, before the sequencing became a viable option. And we know that, looking at the microsatellite data for, say, Brainerd Lakes region, you couldn't really see any structure with these 9 markers. But when you expand out to the thousands of genomic markers, you can start to see those differences between lakes. And so I think there could be a really great opportunity to apply this technique to possibly to these sample sets that already exist, and then integrate the information to basically be able to look at both the ancestral kind of native range of zebra mussels and globally what those populations look like. But at this point, the challenge is getting our hands on the samples, and then of course funding to genotype thousands of more mussels.
Thank you. Okay, next question on the list is wondering what your sampling protocol is for the zebra mussels. Are you sampling from the bottoms, like boat or scuba? Are you scraping from the surface? How are you finding and collecting?
Pretty much however we can get them. We Yeah, we've sort of done all of the above. As I mentioned, we don't really see a lot of structure within lakes, so we don't think it will likely really matter where they come from, if they're pulled off of plants or collected near shore versus, in deeper water. Um, and then we do have some protocols as well for collecting and preserving the samples. So if people are interested in collecting, they should get in touch with us, and we can share those protocols, or even give you a kit that would have some preservative that you could put the mussel in.
Right? All right. Does your lab happen to offer genotyping services for other state agencies?
Yes, yeah. I mean, we actually, this genotyping by sequencing is actually an approach that's widely applied kind of throughout many areas of science. So there are a lot of labs at the University of Minnesota, for instance, that are doing plant breeding and agricultural work that use this to track markers when they breed plants. And so in the Genomic Center here, we actually run this genotyping by sequencing as a service for, dozens and dozens of different organisms. And so, yeah, please just get in touch with me, and I can put you in the right direction to do this for your organism of interest.
Right? All right. Next question, I suspect, spun up from the observation you had from, I think it was Rice Lake, but they're wondering what your thoughts are on inspection programs and if they should focus heavily on the busy accesses, or is it better to focus on where the risk of spread is from a particular water body?
Yeah, that's a good question. And I'll just say that I'm a molecular biologist and geneticist, and so my you know management and is not my area of expertise. But I guess what I would say from the data that we have is that um, we were a little bit surprised to see that some of the lakes that are very, very high traffic, like Mille Lacs and Minnetonka, were responsible for relatively few infestations. Now, part of that could be, the time frame that we were looking. So we need to look, and as we continue to genotype more mussels, we'll see if this observation holds up. But I think a lot of people had presumed that these super high traffic lakes would be sort of super spreaders and responsible for a lot of the infestations, and that really wasn't what we saw in this initial study. And so one way of interpreting that could be that the inspection program is actually working well and doing what it's intended to, right? And that maybe one of the reasons why we're not seeing as much spread from these big lakes is that they do have a lot of inspection effort devoted to them. Um, so I think I personally, I think, one good strategy, if we can get to the point where, each year as new infestations are popping up, we're quickly looking at them, and that's one of the virtues of the technique that Victor talked about, is that help us really close that loop and get this genotyping information as quickly as possible. That could at least let you know, like, are there little areas like Rice Lake or other smaller lakes that are maybe not getting the inspection effort of Minnetonka, that we should be worrying about, and maybe that would be a chance to head off some infestations from those other sources.
Yeah, very interesting. Okay. Next question is wondering if the MDA technology is something that could also be used on things like water samples where they're doing like the eDNA testing for presence.
Yeah, I can maybe take the first stab at it, and then I'll let Victor weigh in. But right now, the MDA is really targeted at a kind of a pure sample. So, the challenge with the eDNA samples is that you have um, you've got kind of a mixture of everything that's in the environment, right? And so um, one couldn't necessarily take like an eDNA sample and then do this sort of genotyping, or at least you would get a mixture that would be much more difficult to interpret. Victor, do you have any other?
No, just to add what Daryl already mentioned, could be possible to apply this, especially for those eDNA false positive results. So in theory, we can enrich our environmental DNA sample, but we have to test that. So it's not a guarantee that all the noise there will be a big problem for our protocol, but that sounds be technically possible.
Perfect. All right. Next question is wondering about potential future expansions for your work, and they're wondering if you're thinking about replicating this to also look at things like quagga mussels, and wondering also if the existing SNP panel if it identify unique lineages of the quagga mussels.
Yeah, I mean, certainly the same approach could be applied to quagga mussels, or we now have golden mussels in California as well, another invasive species. So yeah, certainly one could do that. And we have been talking to, so there really aren't at this point, thankfully, inland invasions of quagga mussels in Minnesota. So they're really confined, to the Great Lakes at this point. But we have been talking to some scientists and agencies out west, where they have quaggas are a bigger issue. Um, but yeah, we currently don't have any plans kind of independently to spin this up for quagga mussels. We're really kind of focused on zebra mussels at the moment.
Sure. Perfect. Okay. Maybe along the similar note of potential expansions, they're wondering if you are considering expanding your work to look at some of the western states where there's an expansion of zebra mussels, potentially with USGS or perhaps other opportunities to expand that work outside of Minnesota.
Yeah, absolutely. That's something we're really interested in, something that we're pursuing. And so we've had some conversations with Colorado's Natural Resources Department, and they have zebra mussels, both zebra mussels and quagga mussels now in the Colorado River, and also some reservoirs and lakes there. So we're working with or hoping that we'll be able to coordinate with that agency to get zebra mussel samples from Colorado. We've also we have some other projects in the lab that are more focused on genetic biocontrol tools for zebra mussels. That work is funded by the Department of Defense. And so we've been connected with the Army Corps of Engineers. And so they have, a lot of issues with zebra mussels on, a lot of the infrastructure that they maintain, like locks and dams and water treatment plants and things like that. And so we're also in the process of working on coordinating with the Army Corps of Engineers to get samples from whatever sites they have all around the country. So that will also help us build out our database. And a lot of those sites are in rivers, a lot of the dams and things like that. And so that's also somewhere where we don't currently have a lot of information. And so I think working with these different groups is going to really uh, help us cast a wider net and expand outside of Minnesota as well.
That sounds great. And Daryl, I hope you don't mind if I'm going to inject maybe my own question as you were talking about the rivers. Do you see are there differences like if you take a point at one spot along a river, for example, something like the Mississippi River, are there differences along the length of the river, or how do you consider like the genetic information when it's in something as geographically dispersed as a river?
Yeah, we haven't seen that yet, but like I said, our sampling has not been very broad. We've essentially sampled the Mississippi from southern Minnesota up to central Minnesota, and at least in that region, you can look at the figure that we have in the paper, and those samples are all homogeneous, at least throughout that region. Of course, Mississippi is much longer than that, so we don't know what it would look like if we went further south, say. Um, so I think it's really an open question at this point.
Cool. Perfect. All right. Next question is wondering if your research would support that the zebra mussel infestation in the Mississippi River is closely related to that in the Illinois River.
I don't think so. I Let me just pull up my slides quick. So we can basically look at those ancestry plots, which will give you a good sense of the relatedness. Um, so yeah, here's the Mississippi River, like I was saying. So we have these five or six different sites throughout the Mississippi. You can see that they all look very, very similar to one another. They have this, kind of green and gray and red dominated ancestry clusters. Um, and then if you look at, I think the Illinois River is on here. Where is that? Maybe it's not on these plots right here. Oh, sorry. It's right next to the Mississippi. Thank you. Yeah, so you can see the Illinois River looks much much more like um, the bulk of Lake Superior and part of Lake Michigan than it does the Mississippi.
Right. All right. Might have time for one or two more questions. Next one is wondering about if you can make some speculations about how zebra mussels are moving based on this. So this person said it looks like maybe it's not all due to high boat traffic. So what are some other mechanisms that might be resulting in a new infestation?
Yeah, that's a good question. I mean, we have like Mike in particular has speculated that some of that local spread could be potentially from movement of equipment. So things like boat ramps that might get
Mm,
shuffled between lakes. But it is really hard, and ultimately, I think we're only seeing the endpoint here, right, with this genetic information, and so it doesn't necessarily let us give us direct insight into what the mechanism of the transport was. We have also had, some of the Western states they do sort of like interdiction of boats as they're traveling, or they'll kind of inspect boats in transit. So we have talked about trying to get samples from those programs as well, which might give us a snapshot at least of what boats are carrying around with them. In terms of other kind of more exotic vectors for spread, uh, there are also, or a couple of years ago, there were also these zebra mussels that were discovered in pet stores. So a number of pet stores were selling these moss balls for aquariums that were found to be carrying zebra mussels with them in many cases. And so we have actually also gotten our hands on some of the samples from those pet store mothball samples that we're going to be taking that we're taking through this genotyping process. And if we see a distinct genetic signature for those, that might give us the ability in the future to see if that actually turns up in lakes, and if there's evidence that potentially the pet trade was also contributing to spread in some cases.
lots of interesting ways to trace things down. All right. So we've got about a minute left. So I think I'm going to move into just this final housekeeping slide. First, thank you, Daryl, and Victor for sharing with us today. Really interesting presentation and kind of fun to see all the kind of different populations and how they might be moving around. I had mentioned earlier that we were recording, so this is a recorded webinar. We do get it put up onto our YouTube channel. The link to that is z.umn.edu/AISTube. It usually takes a week or so for us to get it up there. We take the time to transcribe them, so it takes a little bit of time, but you can watch for it. And you do get an email. Everyone who's registered for the webinar gets an email when that is live and up on the YouTube channel. Also watch for a survey. It should pop up as soon as you leave the webinar, and we'd appreciate it if you take some time to complete that survey for us. I do have Daryl's contact information on the screen in case you needed to reach out to him for anything afterwards. And if you have questions about like the webinar series as a whole or anything like that, our program email is aisdetectors@umn.edu. I see a note from Alex as well that the app should be working now. So if you have that z.umn.edu/mussel strains site from earlier and got that error code, it should have enough capacity to handle folks exploring it now after the webinar. So feel free to browse. Alex says they'd welcome any feedback as it continues to get built out. And I think if I remember correctly, Alex, there's contact information on that page as well. So thank you for everyone for joining us. And again, Daryl and Victor, thanks to both of you for sharing all the information you had for us today.
Thank you.
Great. Bye, everyone.